So our next presentation, also from Science and Engineering, is the wonderful pretty Londhe from Lismore, on development and validation of photoautotrophic microalgal biofilm-based growth systems for high-value product development. I got that one out okay. All right.
So yeah, in short, I'm working on microalgae, and I'm going to extract omega-3 fatty acids from it. My supervisory team consists of these people, and it is an industry project, so I'll be working with an industry. So just to give you a brief idea, I think we all know what microalgae is. We must have seen algal blooms in stagnant waters. And there are some species which are really commercially necessary for us. And I'll be working with 2 of these species to extract omega-3 fatty acids. So microalgae is this very small unicellular microorganism. It's photosynthetic. It plays a very important role in oxygen provision for us. And although there are around 30,000 species, there are quite a lot. There are very small species which have been explored commercially. And since one of my topic is growing of microalgae, so until now they have been grown in open ponds, in closed ponds, in photobioreactors, which is man-made thing. And it's also used for biofuels, for health supplements, and animal feeds and fertilizers. So the microalgal species I'm working on, it produces omega-3 fatty acids. And omega-3 fatty acids, your body cannot produce it. So you have to have it externally. And the species that I'm working on, since they produce it, I'll be working on extraction of these. So until now, microalgae has been grown in racewater plants, in circular ponds, in flat bed photobioreactors. So these are all the different things which we have worked on before, people have developed before. And the problem with suspension-based cultivation system is, first thing is that we used to grow microalgae somewhat like this. So if I give you an example, think of a fish tank, and I'm growing microalgae in the fish tank. And so if I try growing it in it, you would know that enough sunlight would not penetrate underneath because of the depth. So even though scientists have worked on shallowing the depth, it hasn't really worked because the sunlight doesn't reach until down. The nutrient flow is not that good. So until now, in commercial systems, when we are trying to grow algal, we know that around 8,500 liters is pumped, and only 0.1% of the algal biomass is what we need. So you imagine the energy and cost that we need for dewatering and harvesting. So these are the different challenges that we are facing for now. That is light penetration. Like I said, it decreases as you keep going downwards. It's very tough harvesting because all of this is water, and we are looking for biomass, and high capital investment and production costs. So what could we do in order to make this more simple? So my project is growing microalgae instead of suspension-based system, growing it on a surface. So all we could do is just scrap off the microalgae, and this would save a lot of money, a lot of time. So microalgae, they naturally form biofilms. So if you could see the first picture, it's the back of a turtle, and there is biofilm grown on it. So the scientist is just scraping off the biofilm. This is the biofilm grown on a stagnant water, what we usually see. And the last picture is biofilm grown on a ship. So going more into the technical terms, until now this is how we grow biofilm system. And if you can see the diagram E, that is what I am going to work on. There's going to be a substrate and microalgae on top of it, and there'll be nutrient media from down. This is how the microalgae is going to grow. So just to give you a brief review of how this works. So first, we inoculate the microalgae on the substrate, and there's going to be light. Then after that, there's going to be EPS production. So EPS helps in sticking together. So all the biofilm can stick together and a film will be formed. After that, there's going to be biofilm maturation, and then there's going to be detachment after a particular time. So what are the knowledge gaps in my field? So first is there is no proper comparison between suspension culture and perfusion-based. In the two species which I'm going to work on, that is Nanochloropsis and Tyso. We don't know what substrate is the best for the growth, how to optimize different environmental parameters like temperature or pH or different nutrient availabilities, and how can this be scaled. So for now, I'm working in a lab. So we need to see how economical it is to scale it in a commercial system. So it's the same thing. These are the chapters which I'm going to work on. So I have started with chapter 2 for now. This is my 9th month, so it's just the beginning. So first thing is the comparison between suspension and perfusion. So this is what I was working on. The one on the left is suspension-based, and the other is perfusion. So suspension, like I said, how it used to be grown before. So it takes a lot of time. You have to filter it and then dry it, and then you get your biomass. This was quite easy, where you just scrape off the biofilm from it. So I'll just go into brief how I actually did it. I first prepared the nutrient media. So we need a nutrient media for the microalgae to grow. This is how I made it, by sterilizing it. Then after that, you had to set a specific temperature. It was grown in a flask, and then it was filtered, and then it was dried. And then I found out the ash-free dry weight for that. And for biofilm, like I said before, it was collected in small aluminum caps. They were weighed, and then they were dried, and then I found out the ash-free dry weight for that as well. Chapter 3 is going to be finding out different substrates. So I need to work out which substrate will be the best for the microalgal growth and for lipid production, that is omega-3. So we've got different substrates, so I need to find out which is the best. Chapter 4 is going to be optimization of perfused biofilm system. So these were the samples which I was working on. So like I said before, I need to find the best temperature, the best pH, the best nutrient media for this. Yeah, the best substrate type, the best light, the salinity, the pH, and the temperature. And chapter 5 is going to be pilot-scaled perfused biofilm system. So I need to make a bigger system, not only a lab-based system, to check what actually it is to do in a commercial system, because it makes a lot of difference actually working in a lab and then taking out and seeing how it works commercially. So I need to develop an automatic monitoring system for temperature, salinity, pH. Then I'll be extracting the omega-3 fatty acids and check the cost-benefit analysis, also the market potential for the biofilm-based production. And then I need to see what are the challenges and recommendations I get. So the different skills that I learned are aseptic techniques, that is working in laminar flow hood, sterilization, media preparation, inoculation, cell counting, and ash weight determination. And I want to thank my supervisors, the industry, the Australian Algal Services with whom I'm working, and Marine Bioproducts Cooperative Research Center. Thank you.
Oh, thanks, Priti. That is just such a challenging project, a fantastic project. Yeah, you must enjoy it. Although, does it get tedious at times?
It does, because you have to be so precise, and I am not so precise. And then what happens is, even if a small thing gets contaminated, the whole thing gets contaminated, and then you have to redo the project. I mean, redo everything. Yeah, so sometimes it does get tedious, but it's fun.
And do you have to babysit the cultures?
Exactly, yes. So somebody's babysitting them now.
Any questions, Thor?
I'm curious to know, other than the industry support, what inspired you to do this research?
So I remember when I was doing my master's, one of my supervisors was working on this, and we did have a small apple orchard next to our university, and he was growing microalgae to give, I mean, for making it into a fertilizer for that apple orchard. So it was just a side project of his, and he was once showing all of our students, and that's when I saw it for the first time, and it was really fascinating. And I worked with him for 3 months, but I realized that 3 months is a very small time to actually know about it. So I was looking for projects, and I found this, and I came here. Yeah.
With next questions from Banu.
Hey, Priti, really good work, and really amazed by your presentation. Thank you for that one. My quick question is, out of curiosity, that you mentioned you're going to optimize the different, say, temperature, pH, and all. There could be like several combination of everything. So how you decide on selecting the optimal thing? Running the different experiment with different combinations, or?
Yeah. So until now, people have already studied. So we know that the range is between 23 to 25. So then we will have 3 replicates with 23, 3 with 24, 3 with 25, and then also the salinity, and then we have to check different combinations. Yeah, it's going to be a very big setup, but that's what we have thought for now.
Yeah, well, thanks for nicking my question, Banu. I was thinking five variables just for that salinity section. Yeah. That's a very complicated sort of design. Are there any other questions for, Kennedy?
Yeah, just a quick one. Very good presentation, and I love the practicality of it and the benefits of it for sustainability and improved production. Just want to understand, are there any safety concerns or hazards in terms of biohazards, in terms of when you're actually producing these cultures and doing your lab work? And
no, they're not.
No.
Because that's what I'm looking for. The end product is producing omega-3 fatty acids, which is for human consumption. So it has to be actually safe for us to use, so that whatever you extract, you can use it in the future.
Thanks. I have one final question for you. Yeah. So at this point, it's pretty much the lab work leading up to like a small prototype towards some sort of commercial development.
Yeah.
How challenging is that going to be in terms of finding the resources for that, the funding for it at this university and all that sort of stuff? How challenging is building that prototype going to be for you as part of this project?
It's pretty rich, I think. I don't know. Yeah, because I'm with an industry partner. So they are the ones who are funding me, and also yes. MBCRC. So I think MBCRC has got enough funding for this. So it's SCU, it's my industry, and it's MBCRC, all 3 together.
Oh, nice.
So hopefully we've got enough funding for the prototype.
Excellent.
Yeah.
Oh, okay. And well, I was just thinking the space to do it and the expertise.
Yeah, I'm not sure about the space. Yeah, my lab is small. I'm not quite sure how we're going to do it. But since that is my final chapter, which I'll be doing in the third year, maybe we discuss it along the way.
Oh,
Yeah.
well, look, we look forward to having you coming back next year and updating some progress. And
Yeah.
certainly, when you get to the prototype stage, we'd like to have you report back to us on that one too. Thanks.
Yeah, thank you.
Well, look, thanks, Everett. Thanks,
Thanks.
Pretty.
Thank you.